Hello, everyone. I hope you are doing great during this time, and I hope everyone is good during this pandemic time. And wanted to, say my warm welcome to everyone joining us from everywhere around the world. I know from different time zones and different countries. So we are really glad and happy that you joined us today for our certified journal club and our fourth special talk. Today we are going to have Dr. Jon Ashley's presentation today, and Dr. Jon Ashley is currently an assistant professor within the Nanomaterial and Biosensors group at the Department of Healthcare Technology at Denmark Technical University, DTU. He's working on the development of biomimetic receptors for healthcare application, and he undertook a Marie Curie co-fund fellowship at the Barian Nanotechnology Laboratory in Portugal and postdoc position at Cranfield University in the United Kingdom and the University Hospital Basel in Switzerland. He completed his PhD in analytical chemistry at National University of Singapore, and where he was working on the use of non-selects for development of aptamers as receptors for surface plasmon resonance-based immunosensors. His research work is currently funded by a Willem Experiment grant, and we are really honored and I'm excited to have him among us for today's presentation. So, Dr. Jon Ashley, if you want to start the presentation, the stage is yours.
Okay. All right, well, thank you very much. I hope you can hear me properly, because I was playing around with the sound yesterday during the teaching, so I might have messed up my audio settings. So thank you very much for inviting me to come and talk to you today. And it's great that we have these initiatives, online initiatives, to showcase researchers' work. And I hope that you can continue this for a long time. So today I'm going to talk to you about methodologies for rational design and synthesis of biomimetic receptors. In terms of biomimetic receptors, I have classed both MIPs and Aptamers in this class, although Aptamer is sometimes not considered this more natural type of receptor. So first thing we're going to talk about is antibodies. So antibodies are most widely used receptors in research. The market is currently worth 1.3 billion euros per year, and out of that 1.3 billion euro market, about 680 million is spent on antibodies which don't actually work very well. So there are issues with using antibodies as receptors. Firstly, they're used, you need to use animals to raise them. They also have problems with cross-reactivity, batch-to-batch variation, and also often used in the wrong application. And I'm just curious, if you could use the reaction, how many of you have used a antibody before that didn't work in your research? I'm just curious, you could use the little reaction bar to see. I'll have a look through. Is anyone? Not many people then, I guess. So that's interesting. Depends who I ask, really. I mean, if I ask biologists, they tend to get about half hands going up. There's at least someone who's used an antibody which hasn't worked in the past. And so really my research is interested in developing the alternatives to antibodies, in namely the two main sort of alternatives, which are Aptamers, which are nucleic acid based receptors, and molecular imprinted polymers. And I'm quite interested in sort of how these how the molecular recognition works with these two technologies. And the hope is that one day we can actually replace natural antibodies with either of these two technologies. So in terms of applications, I've used MIPs and Aptamers in a number of, in a number of sensors. And so my main research interests is applying these two receptors in biosensing and nanosensors. And you can see lots of examples in the research literature about how these two receptors are used in different types of transducer. But if we do a comparison between the three technologies, so antibodies, MIPs, and Aptamers, we can see a number of differences. I mean, for instance, the way MIPs are developed is completely different to the way Aptamers are developed. In terms of their binding affinities, all three have shown quite high binding affinities towards different targets, although we do have differences between small molecule targets and macromolecules. So for Aptamers, they tend to work well with proteins and cell targets, whereas small molecule targets, they tend to suffer from poor binding affinities. And MIPs is quite interesting because it's the complete opposite, where they say that small molecules tend to have the highest affinity, whereas proteins and cell targets generally are more difficult to produce MIPs for. And when we look at specificity, MIPs often suffer from specificity issues when you're looking at proteins and cell targets. Whereas Aptamers tend to have quite good specificity compared to MIPs. And antibodies can suffer from poor or cross-reactivity as well. And in the case of small molecule targets, antibodies tend to perform quite badly. When we look at stability, though, both Aptamers and MIPs are quite superior compared to antibodies in terms of the stability. Antibodies tend to denature over time, so they're quite sensitive to environmental conditions, whereas MIPs and Aptamers are claimed to have stability for years. When we look at the size, there are differences. Although I've got that the wrong way around. So MIPs have a size of 50 to 100 micrometer, whereas Aptamers tend to be about 3 to 5 nanometer. So it's quite interesting then how these two technologies of MIPs and Aptamers have sort of been parallel to one another, but completely separate. And in this talk, I'm going to show you some examples of where I've used MIPs and Aptamers in using different methods and also in different applications in sensing. So first I'll talk about molecular imprinted polymers. So for those of you who are not familiar with this technology, we basically have a template. And what we do is we mix it with some functional monomers. So these are monomers which have either carboxylic group or amine groups, which form interactions with the template. And we call this the pre-polymerization mixture. We also have a cross-linker to allow for polymer to form. And then upon polymerization, we cross-link our MIP round the template. Now, the difficult part is when we remove the template, we're left with a cavity or recognition site, and upon introduction of the template again, we should get rebinding. And what's really interesting about MIPs is the ability to functionalize them, because we use polymer chemistry, and they come in different forms, from thin films to bulk materials and also nanoparticles. So MIPs can take many different forms and can be functionalized quite easily with different functional abilities. Now, there are a number of different techniques we use to imprint polymers. I'm going to talk about two mainly today. In my group in DTU, we developed, we used core shell imprinting as a technique. So this is where we took magnetic microspheres and we add a silicon layer, and then we form the MIP layer around the silica, and then we had a on-off recognition site. And again, what I want to stress is that these MIPs, or multifunctional MIPs as we call them, are tailorable. So we can form them in a large number of different sort of varieties. In this case, we used it as a fluorescence-based sensor with on-off fluorescence quenching. But because we also had the magnetic core, we were able to separate our magnetic beads. So it was basically an all-in-one nano sensor, which was used to detect antibiotics. But we will talk about that in a bit more detail. So antibiotics, as you know, as one example, the overuse of antibiotics in food has become quite a concern for people. In particular, Denmark is quite protective of its pig industry, pork industry, and the EU regulates the use of these antibiotic residues in livestock, which are destined for slaughter. So there is a need for us to develop sensitive and multifunctional nanomaterials, which could help allow us to detect these antibiotic residues in food samples. So first example of a sensor we developed was for cloxacillin, where we used a magnetic MIP. So we started off simple with just the magnetic core, and we use a thermal solvent method to form these magnetic cores. We then selenate with TOS, and then added our MIP layer. And then in collaboration with Anja Bosen's group in DTU, we developed we used our magnetic MIPs as a sort of food preparation method, or a pre-treatment method. So we were able to add our MIPs, our magnetic MIPs, to our sample. In this case, we used the pig serum, and then we could purify the analyte and concentrate it enough. And then we used a SERS-based sensor with nanopillars, which are up here, as you can see with this SEM. Now, for SERS, basically you form a fingerprint region, which allows us to, which is unique to each analyte, which allows us to perform qualitative analysis. If we add a internal standard, in this case acetic acid we used, we were able to quantify the amount of cloxacillin. So we used our magnetic beads to extract our antibiotic residue from the pig serum. We then deposited it onto our sensor, and we got these fingerprint regions, and we were also able to quantify the amount of cloxacillin that we had, and we established a limit of detection of about 7.8 picomoles. So this was a really nice sort of application of both SIRS and MIPs for a food screening. Another example was to go one step further. We added a fluorescent monomer into our MIP layer. So we mentioned that we formed the magnetic layer again, the magnetic core. We then add the silicon layer. The MPA allows us to add the polymer layer on top. And we added the fluorescent monomer into our mix, and we used fluorescent quenching as our transducer. And when we compare our molecular printed polymer against the control, in this case with molecular printed polymers, they always use a non-imprinted polymer as a control. And you can see that we got a larger rate of quenching for our MIP compared to our fluorescent NIP, and we achieved a limit of detection of 117 nanomolar. We got quite good selectivity out of this nano sensor, and we were able to get a decent recoveries also in pig serum samples. So again, this proved to be quite a good use of multifunctional MIPs as a way of measuring antibiotic residues in pork samples. And this could easily be converted for human serum samples as well. This may be something we'd try in the future. We also developed a multifunctional sensor using carbon dots instead of our fluorescent monomer, because one of the issues with using fluorescent monomers is you get the bleaching effect. So carbon dots are quite attractive because of the fluorescent properties and the lack of bleaching, so they're quite stable. Plus, we're not too concerned about quantum yield. So for other applications of quantum dots, they tend to be concerned about the quantum yield of the carbon dots. But for sensing, I think it's quite a nice application for carbon dots. So the PhD student in our group, Xiao Tong, she developed a multifunctional MIP with carbon dots embedded. Again, we tried it on the deoxycycline as our target, and you can see we've got quite similar results, although she achieved a slightly better limit of detection compared to me. And we also showed that it was quite selective for our deoxycycline compared to the tetraxylene and the other commonly used antibiotics. So in summary, this basically shows you how sort of flexible and easy it is to change the functionality of MIPs to suit your application. And we also showed, well, because of small molecules, we tend to get better selectivity compared to the MIPs for protein targets. Now, as well as small molecules, I have developed MIPs for protein targets. And in particular, I used another type of imprinting technique called solid phase synthesis. And again, I'm trying to point out that we developed here nanoMIPs. So notice how completely changed the functionality of our nanoMIPs. So now we're just developing nano-sized MIPs. So what basically occurs in solid phase synthesis is the target we want to imprint, we will attach it to a solid phase. In this case, it's glass beads. And then we can form a pre-polymerization mixture in a column which contains glass beads. We then can form our MIP around the template, and we can use a temperature wash to remove the nanoMIPs easily from the template. We use the acrylic-based chemistry here because it's water soluble, so it's compatible with proteins. And one of the issues that I didn't mention in the other applications that I showed just before was that one of the challenges is to remove your template after you form the MIP. So this technique is really good because it allows us to remove our product from the template using a temperature wash, and it also allows us to remove all the monomers, unreacted monomers, and low affinity nanoparticles. So it's quite a good way of purifying your high affinity MIPs. The only issues is that the yield is usually quite low using this technique. And this technique was developed by Sergei Polinsky's group and the HEP group in France. And we used this technique in another application. So when I was in Cranfield University, we worked on this Innovate UK project where we developed nanoMIPs against allergens. So milk allergens and allergens, food allergens in general, becoming quite a concern for the Western world, especially food manufacturers, where the regulation and the cleaning and verification of equipment is quite difficult. You can easily get cross-contamination of allergen ingredients. So it makes the controlling of allergen containing ingredients very difficult. And this is why you see the precautionary labeling on food products may contain nuts. And that's really because it's quite difficult to separate in a food manufacturing plant different ingredients. And one area we were focused on was, can we improve upon the cleaning and verification of processing equipment after cleaning using what we call a cleaning in place system. And this would be useful in being able to cut costs in terms of, well, reducing the amount of water, because a lot of food companies, they use a huge amount of water when they wash out their equipment, which is quite wasteful. And so it's really important that we have good analytical techniques for being able to confirm that allergens, that the equipment is allergen free. The only issue is that allergens don't follow this dose-based response. So people who suffer from allergies, you can have very minute amounts of allergens, and that can cause a reaction. So this makes the analysis of allergens even more difficult than it is. So we developed a nanoMIP-based plasmonic sensor. So we use a solid phase synthesis to form nanoMIPs against different proteins, and we concentrate on milk proteins, in particular casein and lactoglobulin. And you can see we basically can treat these nanoMIPs like a protein, so we can conjugate them in the same way to a sensor surface. So hence these nanoMIPs are often referred to as plastic antibodies. And we did a cumulative assay because we had you can have difficulties in regenerating the sensor, although actually our MIPs are acrylamide based, and we also include NIPA as a monomer. And NIPA is a temperature sensitive monomer, so it swells. But I think a lot of people who work in the biomaterials will know this, that you can have these swelling properties. So we could actually use temperature as a way to regenerate the surface of the sensor. And we got quite a decent LOD down below the current level of 2 ppm, which is the level set for ELISAs to detect allergens, so we can beat these commercial kits. And we also tested in wastewater samples, so we took some samples from our collaborator Unilever from their dairy plant, and we spiked in our protein, and we were able to get decent recoveries again. And so nanoMIPs can be treated in the same way as natural antibodies. In fact, you can use the same chemistry to attach them to the surface. And the way we did that was we can use an amine containing functional group in our nanoMIP synthesis. More recently, one of the issues, or a number of the issues that we had with the solid phase is we use a glass bead as our solid support. So problem with using glass beads is that when you attach a template to a glass bead, you can often get abrasion of the target or the template off the beads. Because glass beads obviously rub against each other. That means we can't really stir this sample, the pre-polymerization mixture, so it's quite difficult to get homogeneous nanoMIPs formed. Also, the yields can be quite low, so because you use an affinity selective method. So we decided, because we've been working with microspheres and magnetic microspheres, that we could use this as our stationary phase to attach our template rather than the glass beads. And the advantage of this is, well, firstly, we only need a small amount of support material. The sizes are smaller, so we can actually fit more in. So this is the reason why we can get more yield. We can also attach more protein compared to the glass beads. So that means we're avoiding more of the non-specific products which are formed. And also the yield using our technique, so using the solid phase, magnetic solid phase, gives us a higher yield per gram of microspheres compared to the glass bead method. And because they're magnetic, it's quite easy to separate our MIPs from our template on the magnetic microspheres. And okay, the yield, we use trypsin as our model protein here. So we formed nanoMIPs against the trypsin, and we got an okay KD, although it would be great to get a bit higher. I guess because I used I used SPR to measure the binding affinity. So I used an equilibrium method model, so I ended up getting a bit lower on the affinity. But the selectivity was really good compared to a number of different proteins. And often trypsin's used as a model protein whenever we're developing new imprinting techniques. We would like to expand this to maybe more proteins in the future. Now, I mentioned rational design. So the rational design in MIPs comes in the form of a new technique that we also developed, called using differential scanning fluorimetry. So we used a thermocycler. And basically, this technique has been used in life sciences to measure protein interactions with small molecules. So I kind of came across this technique when I was trying to find an alternative, because our SPR machine was broken in Cranfield University. So I was trying to find another technique that I could use to measure interactions. So I thought about using this. I'd read about differential scanning fluorimetry. And basically how the technique works is that you add a dye, a fluorescent dye. So in this case, we use Cypher orange dye. And basically we can incrementally increase the temperature and measure the fluorescence. So if you have a protein present, so in this case our template, which was beta-lactoglobulin, as we increase the temperature, what happens is the protein starts to unfold, and this sort of exposes what we see as a hydrophobic core of the protein. Now, this Cypher orange dye is specific for the hydrophobic core of the protein, and people don't really know the exact mechanism of the binding, but it seems to bind to the hydrophobic region of our protein. So you can actually use this technique when you have small molecule ligands. So in this case, I prepared the pre-polymerization mixture for our MIPs with our protein. And what tends to happen is when you get interactions of the small molecules with our protein, we get a shift in this folding profile. So we tend to see it as a sort of peak. It could also be a sort of similar to a PCR cycle. And what happens is the small molecule will, as a syngenically bind to the protein in different parts. We get a stability effect, which means that the protein can unfold at a much higher temperature. So people use this technique to measure interactions of small molecules with proteins. And so I thought maybe we could use this as a way of rationally designing our nanoMIPs. So you can see here we have different binding profiles or melting profiles. And TBE, so we use different types of monomers. So hydrophobic monomers such as TBA tend to destabilize our protein, causing it to unfold more quickly, whereas hydrophilic monomers such as acrylic acid tend to stabilize it more, and we get a higher sort of binding effect. So we could actually use this technique to design the monomers that we should put into our pre-polymerization mixtures. Because one thing I didn't mention in the previous examples is the time taken to find the right ratio of monomers cross-linkers to template, which often is done in a trial and error approach. So this method allows us to basically high throughput the design of our nanoMIPs. And you can also use it to characterize the protein binding of the nanoMIPs, which is characteristic of this sort of aggregating protein. So there is a question of whether when we do protein imprinting, how the protein, how stable it is and how does it unbind during the polymerization. And it does suggest that without surfactants, the nanoMIPs, the protein targets tend to unfold. And you can compare this to the native protein on its own and our pre-polymerization mixture with BLG. And we can see also that the nanoMIP doesn't show any profile or signal from our separate orange. So we know that this signal is due to the protein only. And so we also looked at the effect of SDS. Now SDS at high concentrations tends to cause the protein to unfold and denature, hence page tail, but at much lower concentrations it tends to stabilize the protein. So when we're imprinting, we notice a complete difference in the nanoMIP when we add a surfactant. So we can actually use this technique to design our nanoMIPs and the surfactants and the additives that we use in this technique. So it really can help to improve and speed up the development of the MIPs. So in summary, I talked about coarse shell imprinting, which gives you the high functionality and tailorability, which makes it very useful when we're designing all-in-one nano materials. We talked about solid phase synthesis, which allows us to remove our MIPs easily from our template. We improved upon the solid phase by using a different imprinting or solid phase, which seems to improve the yields, and we can get a more homogeneous sample. And we also talked about a different rational design approach to forming protein binding nanoMIPs. So now I'm going to talk about Aptamers. So I've been quite lucky in the fact that with Aptamers, during my postdocs and my PhD, that I've had the opportunity to develop both technologies. So I have a good idea of both molecular imprinting and selects. And it's quite interesting how these two technologies, MIPs and Aptamers, are in parallel, but they don't really mix at all. I mean, there has been attempts to hybridize the two technologies. So you have these hybrid Aptamer MIPs, although it hasn't really been that much development in that area. Mainly because you can't really, it's difficult to justify mixing these two technologies together. So Aptamers are completely different because we use a biological approach to develop them. The MIPs, we use chemistry and polymerization, whereas Aptamers, we use a combinatorial approach. So Aptamers are basically single-stranded DNA or RNA, less than 100 nucleotides in length and often fixed length. And they fold into these three-dimensional structures. And when we mix them with a complex, we get interactions in the same way that we get with the MIPs, although you can see that the MIPs has this sort of cavity shape, whereas an Aptamer kind of wraps round or fits into the Aptamer. It's a lot more flexible and it's a lot more sensitive to the buffer conditions. Now, Aptamers are selected using the select, which was developed by Ellington and got Larry in the US about 2 decades ago. And the process often involves using an initial Aptamer library and mixing it with our template or target in this case. Notice the change in terminology. When we use it, we talk about aptamers, we talk about target rather than a template. We then can separate all the sequences of DNA which are bound from the unbound DNA. We get rid of all the unbound DNA, and then we do PCR to amplify those sequences which bind, and then we repeat. So we often use a fixed length DNA libraries. The number of rounds varies between targets, so between 5 to 20 rounds of selection are needed. So it's often based on the target you use. And because we keep repeating the PCR step, we get what we call PCR bias, which means that aptamers can be selected on their ability to be amplified using PCR rather than their affinity. So this is one of the major issues with Select's technology, is that often we do our sequencing and we get a number of sequences, but often our sequences with the highest copy number is not necessarily the one with the highest binding affinity. And this procedure is time consuming and costly, so it takes a long time to develop these aptamers compared to the MIPs, although MIPs in its own right can take some time to get the interactions just right. This one takes even longer. So here's an example, because I'm in the healthcare department, but I think we used to be in nanotech, so a lot of the examples I've shown before have been food orientated. So here's an example of where I've worked in the biomedicine. So DUX4 is a transcription factor, which is being implicated in FSHD muscular dystrophy, which is a chronic and terminal sort of disease, progressive disease, which causes muscle wastage in patients. And recently, they found that this transcription factor DUX4 has been implicated in the progression of this disease. So basically, the gene in healthy individuals, the gene that expresses DUX4 is inactive, so it doesn't express this protein, but in patients we get overexpression of this transcription factor, and this leads on to the progressive muscle wasting. So one of the therapeutic possibilities here is to develop an aptamer. So we developed a single-stranded DNA aptamer. I did this in my first postdoc, so I did the selection, although the actual paper was only released this year. So it did, as I mentioned before, it does take a long time to develop aptamers. And you can see this is the top sort of aptamer sequence that was found, and it had a KD of 20 nanomolar. Although it's worth noting that transcription factors are often the easiest targets to develop aptamers against, because they have a predisposed ability to bind to DNA. So often when you select aptamers against transcription factors, they often mimic the double-stranded DNA or DNA region that the transcription factor usually binds to. And we use fluorescence polarization to get the KD value. And what's really good is that eventually we reached the point where we actually did the crystallography study, and we were able to characterize the binding interactions of the aptamer, and we found that this triple C bulge was primarily involved in the interactions with these arginine groups on the DUX4 protein. So it's usually quite unusual to get to this point where we've actually shown the mode of binding of the aptamer. So as I said, it takes a long time to develop aptamers, and it's kind of the rationale for my current research is I wanted to make aptamers more accessible to groups to try and rival antibodies more. During my PhD, I used a non-evolutionary method to select aptamers using a special technique called capillary electrophoresis, which is basically a glorified page gel with a detector at the end. So it's a bit like in fact, if you have a bioanalyzer in your lab, it's exactly the same technology. And you have a fluorescent detector. And you can actually use this method as your way of separating your bound DNA from your unbound DNA. So in 2006, the Kylov group in Canada developed this non-select group protocol where they could avoid the amplification step and just do continuous partitioning. Because capillary electrophoresis is quite a sensitive technique when you match it with laser-induced fluorescence detection. And you can see here that you can actually quantify and show the collection window and collect the bound DNA. So your protein migrates at a certain point, and then your DNA migrates later. And then in between, we should get complex peaks if you're lucky. Although in this particular case, one of the issues with this technique is that the capillaries, you need very small samples. So imagine your page gel, you often can load a lot higher volumes of aptamers. But with a capillary, you can only inject nanoliters of samples. So this sort of limits the amount of library that you can screen. But still, this technology shows that actually maybe the number of rounds or selects rounds that you're needed, or this repeated rounds of selection, actually aren't really needed at all. So I'm kind of focused more on developing these non-evolutionary techniques where we don't have to do the amplification step, and hence we remove the need for PCR bias. So with my, or the project I've just finished, I was sort of inspired by a paper I read in 2017, where they talked about terminal deoxynucleotidal transferase, which is quite an interesting enzyme. And what's unique about it is it's a polymerase enzyme, which doesn't need a template DNA in order to amplify DNA. So what it does, this enzyme can basically add nucleotides stepwise to a oligonucleotide or initiator sequence, and it can be done randomly. So it will randomly incorporate nucleotides. And what's important is the metal line used that can control a kinetic bias, because often the TDT has a bias for certain nucleotides, although we can adjust the mixture to account for this. And it's supposed to form it's what we call a living polymerization, which basically, and this is why I was interested, because from a chemistry point of view, living polymerizations are quite interested in MIPs as well, because with polymers it's very difficult to control size distribution. And the living polymerization is a synthetic method which really allows you to get narrow size distributions of polymers. So I thought, well, maybe this enzyme could be used in formation of libraries of DNA. And would it be better for our selects methodology? And so I formed some reactions with different mixtures of nucleotides and see what came out, and sort of optimized the buffer conditions, tried different things. Now, if you're squeamish about unconventional gels, then I would look away now, because this is what I got. So I got like a DNA smear, and this is what I wasn't really expecting. This, what I was expecting was some narrow band, and the reason being is this original paper, they only used poly T initiator sequence, and they only used DTP. TTP. So they minimize the amount of secondary structure that was formed during the polymer growth. So actually, this living polymerization mechanism that this enzyme is believed to go by doesn't work when we have a mixture of nucleotides, because what happens is we get secondary structures formed which inhibits the growth of the chains of DNA, and so we get this size distribution or smear. And usually when biologists get this type of gel, they think that the DNA is degraded, but because this is a recombinant DNA, and we've looked at the possibility that we're overloading the sample, we still get smear even when we decrease our DNA concentration. And you can see that we can control the size distribution of our DNA based on the time. So longer times we get larger size distributions. And you can see I've actually ran out of single-stranded DNA ladder. It's one of the issues that they don't actually sell single-stranded DNA ladders that go above 100 nucleotides. Although you can use a double-stranded DNA ladder, but often doesn't resolve very well in the gel, especially at the 5%. At a 20% gel, it would resolve quite nicely. And so, well, I thought, well, maybe this will aid our selection. So if we use these libraries rather than a fixed length DNA library, we could possibly be able to do this in a non-evolutionary methodology, because we're basically increasing the efficiency of our selection. And this is just to mention TDT. It came from its role it plays an active role in immunology, so it fits in with the theme of our department. Yeah, I will just move. So what we did then was to develop a new methodology, and instead of using the fixed length libraries of DNA, because one of the reasons why we need to do these continuous rounds of selection is because often the amount of DNA which binds in a fixed length library is quite small, and often we can't quantify it enough. Whereas if you have a large size distribution, you're actually starting to screen your DNA based on both sequence and length. So you're actually increasing the efficiency of the selects process. And so what we did was we formed the libraries of our TDT, as I showed in the last slide. We incubate it with our target and separate the bound DNA from the unbound DNA. And I'll show you on the next slides that the population of our binding DNA is quite high. We then have to convert it into double-stranded DNA so we can sequence. So we often, in this case, are treated like an RNA molecule by adding a poly A tail, and luckily we can use TDT to do this again. So we use the TDT twice to form our poly A tail. We can then use a poly T adapter reverse primer in order to do PCR to convert it. And then we use next gen sequencing. And from our next gen sequencing, we can actually easily identify the random region of our aptamers. So these are the initial results. So as I mentioned before, we saw this denaturing gel where we get our size distribution. So we can tailor the size based on the reaction time. And we can perform a EMSA or native gel to separate the protein complex, DNA which binds to the protein. In this case, we use thrombin because it's a model protein to use for new methodologies. And you can see that as we increase the size of our DNA library, we start losing our resolution. So we have this sort of double-edged sword of we can't grow our library too much, although we can use maybe agarose gels to separate, or capillary electrophoresis if I had access to that machine, which I don't at the moment. Then you can then you have to be careful that you don't extract. So you could do a simple gel extraction to take the complex out and then do the next steps. We also developed an aptamer against the Lactoferrin, which was a urine biomarker for urinary tract infection. And you can see here that we also get a complex peak formed quite quickly. And what's interesting is we're using a stain here. We also do our, we show that the stain doesn't bind to any protein, so it's purely DNA that we are visualizing here, and complexes. And we also, and this is where the rational design comes in, we can also tailor very easily nucleotide ratios. So thrombin has been characterized before as aptamers have been developed against it. So you can show how the binding changes with different ratios of nucleotides that you put in your mixtures before you form your libraries. And we knew that from thrombin that G quadruplexes are quite important in the binding to thrombin. So we were able to tailor our nucleotide mixtures to the targets. We also can show that it works over different buffer conditions. So there's a lot of this is in a sort of similar way with MIPs, where you have to change your reaction mixtures and stuff like that. So eventually we did the NGS on both samples from them. They both showed quite G-rich because we deliberately made G-rich. So G quadruplexes should feature quite heavily in our sequences that we found, although our copy numbers were quite low because of we needed to really optimize the NGS more. We had switching, structural switching. We did lose a few aptamers, but luckily you only really need 10 to 20 sequences anyway because before NGS was around, we had normal Sanger sequencing. And you can see Lactoferrin also heavily featured G quadruplexes in the binding motifs. So this is and actually it's worth pointing out that in most successful aptamer sequences G, they often are G-rich in nature. So this is quite promising in terms of the NGS results. You can also see we found sequences of large size distributions based on the random region. So for thrombin, these were the top two sort of sequences that we focused on because of the delta G. So we look at the secondary structures, and often what you're looking for is a high delta G value, Gibbs free energy, which means that this is a stable conformation. Although it's worth noting that actually three-dimensional binding is quite important in Aptamer characterization, and the Unifold software only shows you secondary binding structures. So what happens is that Unifold isn't particularly good at actually predicting good Aptamer binders, and we may be missing a lot of binders through based on this. So I also included what we call G-score. So I used a program to predict in each sequence how likely it is to form quadruplex G sequences. And what we found is with the TH10, we had two possible G quadruplex sites. And this is quite interesting because it means that we have the possibility here that we've selected a bivalent Aptamer, because we can screen much larger sizes of DNA. We also had the smaller Aptamer. So these were the top two based on the SPR studies, which I'll show you in a sec. And so we did SPR of these top two Aptamers, and you can see we got quite a good binding affinity for the bivalent TH10, although we did, compared to some other bivalent engineered TH thrombin Aptamers, this is a little bit higher. They were in the picomolar range, but still quite good result because we actually selected this one rather than a trial and error approach of conjugating to Aptamers, because thrombin is known that the Aptamer, there's two Aptamers being developed against it, which bind different sites on the thrombin. So there's actually two binding sites. So we're quite certain that we have the binding, bivalent binding. Although I use both 1-to-1 model and 2-to-1 model using SPR to determine the binding affinity. And then we also showed the selectivity of TH10T we took as the lead candidate, and we can see that it showed quite good binding compared to thrombin, compared to the other types of protein which you might find in blood, because thrombin is a protein found in blood plasma, which causes clotting. For human lactoferrin, we found these three Aptamer sequences, and they were a lot larger. And they also showed quite a large potential for G quadruplex sequences, although we cannot say for certain if the exact binding motif is for these Aptamers. And so we also did our SPR binding, and lactoferrin showed quite a strong binding. So we were getting down into quite low nanomolar range. Again, we used the 2-to-1 model and the 1-to-1 model just to see, on the assumption that larger sequences of DNA might have multiple binding sites. So we also tested the binding against HSA as the highest protein which would be found in urine samples with as a sort of in patients with the urinary tract infection. So our next step now is to develop, put this in the impedance sensor. So we're currently developing that in our group. And so in summary, what I can say is we've developed a new non-evolutionary screening technique. So it's worth noting this was a one-round selection. So we've reduced the time and the resources of selects. And we've taken advantage of these broad size distributions of libraries that have been formed using TDT enzyme, which has increased the efficiency of the binding and the population of aptamers which actually bind. And we've also been able to tune the apparent binding, so we can change a lot of things. There's a lot of flexibility in the formation of our libraries. We've been able to convert these libraries into double-stranded DNA through the sequencing, and we've confirmed the binding of 2 targets, protein targets. And what's also interesting, when it raises the possibility of what the role of TDT was in the immune system. So I was always wondering whether TDT may have been had a different role as instead of forming receptors in T cells or lymphocytes, maybe single-stranded DNA formed the basis of early adaptive immune systems. Although that's an answer that hopefully someone else can answer. And so with that, I'd like to just thank the funding that we got, so the Marie Curie co-fund and the Willem experiment, for allowing me to do a risky project, which had no guarantee of success. Also, the molecular printed polymers research was funded by Willem Foundation through my group, my boss's group, and the Innovate UK project, which was focused on the allergen detection project. And I guess I'll finish with a political statement, as you should do. I just wanted to demonstrate that in my postdoc career, I've actually used freedom of movement quite well until Brexit came along, and without freedom of movement, I wouldn't have been able to, maybe I would, but it would be a lot harder for me to move around Europe. And the fact that I can gain this research experience and develop my research, I'd like to thank people in these groups. So I started my first postdoc in Basel with Michael Simreich and Jochen Kinter, were supervising the project on the Aptamers for Talk4. And then when I moved to Cranfield, where I worked on MIPs, developing MIPs with Sam Tottle and Eunice, and we had collaborators Roy and Linda and from Manchester University, and Thomas as well, which I've forgotten to add on this slide. And when we had moved to DTU, I had Sun, who was my boss, and collaborators Anya Boson and the team. And I also like to thank Anna Johansson and Paolo for helping with the NGS for this current research project. And I also did a year in INL in Portugal, where I was working under Martha Prado at the start of this Aptamer selection methodology. And with that, I'd like to thank you for your attention and ask if there's any questions.
Yeah, thank you so much, Dr. Ashley. That was fantastic presentation. Thank you so much.